It's 2024, and most of us has something like this: a vaccination card. Ever since the pandemic began in 2020, we are told to be vaccinated in order to prevent the spread of severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2, which is the virus that caused COVID-19. It worked. It prevented the spread of the virus in our bodies worldwide. But how and why? It is all thanks to T cells. What are these cells, and how does it recognize and respond to pathogens? Well, the key component of T cells that grants them these abilities are their T-cell receptors. It all began in the 1940s when a scientist named Jacques Miller discovered that plasma cells were responsible for producing antibodies, setting the stage for exploring our immune system. Fast forward to the 1950s, Bruce Glick and Timothy Chang performed a unique experiment on chickens. They removed a small organ called the bursa, and to their surprise, the chickens could no longer produce antibodies. This led to the discovery of B cells, essential for immunity. But there was still a missing piece. In the 1960s, Jacques Miller again made a breakthrough by removing the thymus from mice. The mice became severely immunocompromised, unable to fight infections or reject skin grafts. This led to the discovery of T cells, named after the thymus. But the big question remained: how did these T cells recognize threats? Finally, in the 1980s, scientists like Alan Harris and Jim Godding uncovered the secret: T-cell receptors. These receptors, unique to each T cell, allow them to recognize specific invaders. And with this discovery, the mysteries of how our immune system precisely targets the threats began to unravel.
T lymphocytes or T cells are a type of white blood cells that is crucial to the adaptive immune response, which are then divided into several distinct types, each with specific rules and features. Cytotoxic T cells recognize the respond to specific antigens represented by MHC class I molecules, which are displayed on nearly nucleated cells throughout the body. Effector CD8+ T cells, primarily responsible for immediate and effective responses to infections and malignancies despite having short lifespan. Memory CD8+ T cells, generated from a subset and activated effector T cells that survive post-infection. They possess the ability to self-renew and can persist for years, providing long-term immunity. Helper T cells, or the CD4+ T cells. These cells coordinate the immune response by activating other immune cells, such as B cells and macrophages. And instead of directly killing infected cells, they secrete cytokines that boost the overall immune response. Regulatory T cells. These cells preserve immune tolerance by regulating and suppressing excessive immune response that might cause autoimmunity, thus preventing other T cells from attacking the body's own tissue. Natural killer T cells. These cells connect to the innate and adaptive immune system by recognizing lipid antigens presented by CD1D molecules as opposed to the classical peptide antigens. External environment or cues are detected through the interaction of T cells receptors with an MHC antigenic peptide complex, which are then converted into multiple signal transduction pathways within the cell, leading to the activation of various genes with the assistance of specific transcription factors. The core TCR complex consists of multiple components that collaborate to recognize the antigens and signal the T cells to initiate response. TCR chains. The core of the TCR is made up of two protein chains, usually called TCR alpha and TCR beta, or in some cases, TCR gamma and TCR delta. These chains form a pair and include regions capable in specifically recognizing antigens presented by other cells, with the tips of these chains featuring variable regions that determine their specificity for different antigens. Cluster of differentiation tree, or the CD3 chains. Surrounding the TCR chains are 6 proteins collectively known as the CD3 complex, which include the CD3 epsilon, CD3 gamma, CD3 delta, and CD3 zeta chains. These proteins do not directly recognize antigens but are essential for signaling, which the TCR binds to an antigen. The CD3 proteins rely signals inside the T cells to activate it. Co-receptor CD4 and CD8. These molecules are not part of TCR complex but function in conjunction with it. CD4, typically present in helper T cells, aids the TCR recognizing antigens presented by MHC class II molecules, while CD8 found on cytotoxic T cells help in recognizing antigens presented by MHC class I molecules. Signaling molecules. The TCR complex is linked with various molecules including the kinases like lipocyte-specific protein tyrosine kinase and adaptor proteins such as CD3 chains, which facilitate the transmission and activation signal from TCR into the cell to trigger an immune response. These components of the TCR complex collaborate to enable T cells to recognize the specific antigens and initiate an appropriate immune response, including effector functions such as cytotoxicity and cytokine production. MHC molecules are vital components of the immune system, enabling T cells to recognize and respond to antigens. They are categorized into two main classes, MHC class I and MHC class II. MHC class I molecules present endogenous peptides, which are derived from proteins produced within the cell, to CD8 plus cytotoxic T lipocytes. In contrast, MHC class II molecules present exogenous peptide originating from extracellular proteins to CD4 plus helper T lymphocytes. This interaction is crucial for activating virus immune cells, including B cells and macrophages, thereby coordinating a comprehensive immune response. A homodimer consists of two identical monomers, and in the context of TCRs, an example is the CD8 co-receptors, which can form homodimers like CD8-alpha, CD8-alpha. This dimerization is essential for specific signaling processes in T cells. In contrast, heterodimer consists of two different monomers, such as the combination of TCR-alpha and TCR-beta chains in TCRs. This heterodimerization forms the TCR complex, crucial for recognizing antigens and enabling a diverse range of antigen recognition and specificity in most mature T cells.
A primary function of TCRs is antigen specificity, which allows T cell to recognize particular antigen presented on the surface of other cells, whether they are infected by a pathogen or have become cancerous. Once the T cell recognizes the specific antigen, it can orchestrate an appropriate immune response, such as directing cytotoxic T cells to eliminate infective or cancerous cells and prompting helper T cells to produce cytokines that coordinate a broader immune reaction. In general, TCRs identify specific peptide fragments of antigen presented on MHC molecules. T-cell receptors recognize both the peptide component and the MHC molecule presenting it, exhibiting dual specificity. The characteristic is known as MHC restriction, which ensures that T cells only respond to antigen presented by their own body cells. Upon binding the peptide MHC complex, TCRs initiate a series of intracellular signaling cascades through associated proteins like CD3 and zeta chains, which is part of signal transduction of TCRs. These proteins are essential for transmitting signals to lead T cell activation, proliferation, and differentiation into effector cells. T cells express accessory molecules that enhance the binding strength to MHC molecules and provide additional signals necessary for full activation. During adaptive immune responses, T-cell receptors play a pivotal role in recognition of antigens. They specifically recognize and bind to peptide fragments of antigens presented by major histocompatibility complex or MHC molecules on the surface of antigen-presenting cells or APCs. This interaction is crucial for the activation of T cells. Each TCR is specific not only for a particular peptide but also for the MHC molecule presenting the peptide, and thus, this dual specificity ensures that T cells can effectively monitor the internal environment of body cells for signs of infections or malignancy. When T cells encounter antigens through the T-cell receptor, they relay information about the quantity and quality of antigen engagement to the intracellular signaling machinery, which then initiates a series of signaling cascades that lead to T-cell activation and its effector functions. The TCR lacks a substantial intracellular domain, so it associates with CD3 molecules, which have intracellular signaling domains that connect the TCR or CD3 complex to the downstream signaling machinery. ITAMs, or immunoreceptor tyrosine-based activation motifs, are specific sequences found in the CD3 and zeta chains of the TCR complex. During TCR activation, these motifs undergo phosphorylation, which enables them to serve as docking sites for various signaling proteins. This phosphorylation is a crucial step in initiating and propagating the signaling pathways that lead to T-cell activation and function. Activation begins when a naive T-cell encounters a peptide MHC complex that matches its specific TCR. The binding of the TCR to the peptide MHC complex initiates a series of intracellular signaling cascades, primarily through associated CD3 molecules, which are essential for transmitting activation signals into the T cell. This process often requires additional signals from co-receptors, such as CD4 or CD8, which stabilize the interaction and enhance the strength of the signal. Once activated, TCR signaling leads to various cellular responses, including T cell proliferation, differentiation into effector and memory T cells, and the production of cytokines. The strength and duration of TCR signaling are crucial for determining the fate of the T cell. Strong signals can promote differentiation into effector cells capable of directly killing infected cells or helping other immune cells, while weaker signals may lead to energy or tolerance.
Conventional T cells identified by the presence of CD4 and CD8. Cell surface receptors utilize clonally diverse T-cell receptors or TCRs to detect antigens originating from pathogen proteins presented as peptide fragments bound to major histocompatibility complex or MHC molecules. T-cell reproductions play a crucial role in identifying the antigenic elements of vaccines and starting the adaptive immune response, as they can recognize a broad spectrum of peptide fragments from pathogens or cancer-related mutated proteins. The T-cell reproduction's capacity to identify peptides displayed by the MHC molecules is essential for detecting intracellular antigens that antibodies cannot access.
A vaccine is a biological agent designed to safely trigger an immune response that provides protection against infection or disease upon future exposure to a pathogen. To achieve this, the vaccine must include antigens that are either sourced from the pathogen or synthetically created to mimic its components. Once injected into the muscle, the vaccine's protein antigen is absorbed by the dendritic cells. These cells are activated through pattern recognition receptors or PRRS in response to danger signals in the adjuvant and subsequently migrate to the draining lymph node. Peptides from the vaccine protein antigen presented by MHC molecules on dendritic cells activated T cells via their T-cell receptor or TCR, along with signaling from soluble antigen through the B-cell receptor or BCR. This activation promotes B-cell development in the lymph node. T-cell dependent B-cell development leads to the maturation of the antibody response, enhancing antibody affinity and generating various antibody isotypes. Short-lived plasma cells produced a rapid increase in serum antibody levels over the next 2 weeks by secreting antibodies specific to the vaccine protein. Additionally, memory B cells are generated, which help maintain immune memory. Long-lived plasma cells migrate to bone marrow niches, where they can continue to produce antibodies for decades. T-cell receptors are crucial for the success of vaccination. As well, they allow T cells to identify and respond to specific antigens presented by major histocompatibility complex or MHC molecules. Examining the dynamics of the TCR repertoire offers insights into vaccine efficacy. By evaluating the diversity and specificity of TCRs before and after vaccination, researchers can gauge how effectively a vaccine stimulates a targeted immune response.
T-cell receptors, known as TCRs, are crucial to the adaptive immune system, allowing T-cells to recognize antigen presented by the MHC molecules on infected or abnormal cells. This antigen specificity enables targeted immune responses, including the activation of differentiations of T-cells into various subtypes, such as cytotoxic, helper, and regulatory T-cells. TCRs also play a role in maintaining immune tolerance, preventing autoimmunity, and ensuring effective defense against pathogens. In vaccinations, TCRs are key to developing long-term immunity. Vaccines introduce specific antigens that TCRs on CD4 plus and CD8 plus T cells recognize, prompting clonal expansion and differentiation into effector and memory T cells. Memory T cells enable rapid and robust responses upon subsequent exposures to the same pathogens. Thus, TCRs are essential not only for immediate immune defense but also for the lasting protection provided by vaccines.